Complex 1 of the electron transport chain, that is NADH dehydrogenase, is a large multi-subunit complex with about 40 polypeptide chains. It contains one molecule of FMN, that is flavin mononucleotide, and 6 to 7 iron sulfur clusters, and these participate in the electron transport process. Complex 1 passes electrons from NADH to coenzyme Q. Hydride ion of the NADH is removed and is converted into proton and 2 electrons. During the transport of each pair of electron from NADH to coenzyme Q, complex 1 pumps 4 protons across the inner mitochondrial membrane. Iron sulfur centers are non-heme iron complex to sulfur. These are mainly of two types, as shown in the figure. These iron sulfur centers consists of equal numbers of iron and sulfide ions, and both are coordinated to 4 cysteine sulfhydryl groups. Complex 2 consists of an inner mitochondrial membrane bound enzyme, succinate dehydrogenase. This enzyme is a part of Krebs cycle where it converts succinate to fumarate. In the process, 2 electrons are released. These 2 electrons are first transferred to FAD and then to iron sulfur center of the complex 2 and finally to coenzyme Q. Complex 2 does not pump protons during the transport of electrons across the inner mitochondrial membrane, unlike the complex 1. Coenzyme Q, also known as ubiquinone, is a benzoquinone linked to a number of isoprene units. It is named ubiquinone because of the ubiquitous nature of the quinone. Coenzyme Q has 3 redox states: fully oxidized, that is ubiquinone, Q; semiquinone, or also known as UB semiquinone; and fully reduced state, that is ubiquinol, QH2. Coenzyme Q is the only electron carrier in the electron transport chain that is not a protein-bound prosthetic group. It is a carrier of hydrogen atoms, that is protons, and electrons. It draws electrons into the respiratory chain, not only from NADH but also from FADH2.